So there are various models, methods, and phenotypes in studying behavior. Genetic models for inheritance and behavior. There's a single gene with a dominant or recessive pattern of inheritance. These are very rare. Most behavior genes are more complex than just dominant recessive. There are some examples: Fragile X syndrome, Huntington's disease, Lesch-Nyhan syndrome. There's the polygenic additive model. We talked about a polygenic trait is a trait that is controlled by more than one gene. Epistasis, we also talked about, if you have a certain phenotype, it will block the rest of the phenotypes from developing. That is part of the polygenic additive model. There are complex traits, which most of our behavior traits are very complex. They're controlled by several genes and environmental influences. And heritability is used to measure environmental effects on behavior. It's a scale from 0 to 5, I think. 0 to 10. We talked about the heritability before, but the higher it is, the more likely it is that these things are controlled by genes and passed on through generations. Genome-wide association studies, or GWAS for short, are used to search for associations between a behavioral trait and combinations of a single nucleotide polymorphism, or a SNP, also called haplotypes. We've talked about SNPs and haplotypes several times this semester. Using the GWAS, scientists are able to scan thousands or tens of thousands of genomes to see whether a specific haplotype is found significantly more in people with a particular behavior, maybe like schizophrenia, than in the general population. If so, then a gene associated with this behavior may be the same chromosome region as the SNP. This chromosome region can be studied in detail, searching for genes that may contribute to the behavior being studied. So, like I said, most behavioral genes are very complex. It's not just the genes, it's not just dominant recessive. It's the additive properties of adding all these genes together, plus the environmental influences. Are these genes going to be turned on or off depending on what's going on around you? Behavior. The definition of behavior is a reaction to stimuli that alters an organism's response to their environment. To study behavior, scientists have to assign a phenotype to that behavior. It can be innate, or something you're born with, or learned, something you gain with experience. The definition of a behavioral phenotype must be precise enough to distinguish it from all other behaviors and from the behavior of the control group. But there will always be variation. There will always be exceptions to the rule. In science, anything you talk about, there are always exceptions to the rule. Many disorders with a behavioral component are associated with alterations in the structure and or function of the nervous system, your brain, your hormones. That's what controls a lot of our behaviors. The nervous system receives and processes stimuli. Stimuli can be simple or complex, involving many senses, memory, and innate and learned behaviors. two components of the nervous system. This should be reviewed. You should have seen the nervous system at some point. The brain and spinal cord is called the central nervous system. That's what controls everything. And then your peripheral nervous system is all the nerves that shoot out to the rest of your body. That's all the green in the picture. You also have other sense organs. Your skin is a sense organ. There's a lot of sensory nerves that receive stimuli in your skin. Your nose, your ears, your eyes, those are all sense organs that control your senses. Synapses are the functional connection between neurons. Neurons are the official name for a nerve cell. And you can see at the bottom, I can annotate it. The synapse is the actual space between the neurons. And then when we talk about the presynaptic neuron, that's the one before, just like when you read left to right. And the postsynaptic neuron is the one after the synapse. So nerve or electrical impulses travel from the presynaptic neuron. The impulse stimulates the release of chemicals called neurotransmitters across the synapse, and to attach to receptors on the postsynaptic neuron and start the next electrical nerve impulse. This is just the general way that your nervous system works all the time. So the signal comes in to the presynaptic neuron, and then it travels, and then something happens in this little space we'll see in a minute. And it goes to the postsynaptic neuron all the way. And then you have another one down here, another one over here, another one over here. And the same thing keeps happening among thousands and thousands and thousands of neurons all the time in your body. So this is your neuron, the signal travels down, and then eventually goes to the next one. And this is the synapse up close. So these little vesicles with neurotransmitters in them. The signal comes down, stimulates the neurotransmitters to release into the synapse, go to the next neuron, and start the signal there. Well, this is the way everybody's neurons work. They're supposed to work this way all of the time. Sometimes they don't, and that's when problems are caused. So variations in the timing and duration of neurotransmitter activity can affect behavior. There's some neurotransmitters and the processes they affect. In your body, you have one called acetylcholine. That's for muscle stimulation, heart rate, learning, sleep. Dopamine is your happy neurotransmitter. That one is for learning, memory, mood, positive reinforcement. GABA is another neurotransmitter. It's an inhibitory transmitter. Inhibit means to stop. So it is a neurotransmitter that stops something that is supposed to be stopped. Serotonin is another one that controls your sleep, mood, appetite, pain, body temperature. So neurotransmitters control all these different functions in your bodies. Many recreational drugs mimic neurotransmitter action. Cocaine, morphine, heroin mimic your endorphins. Nicotine also mimics endorphins. Amphetamines, like meth, mimics norepinephrine or epinephrine. That's the neurotransmitter that gets a person all pumped up and excited. Normal brain function depends on normal synaptic behavior. Like I said, those GIFs that we were looking at earlier, that is normal synaptic behavior. That is the way your nervous system is supposed to work all the time. There are behavioral disorders that are associated with mutations in genes affecting synapse function, whether it's the release of the neurotransmitters, the timing of how long it takes to get across the synapse, or a lot of the time it's the receptors on the second one are mutated. You can see there's a lot of behavioral disorders associated with synaptic defects: Alzheimer's, alcohol sensitivity, autism, bipolar, Huntington's, depressive syndrome, Parkinson's, schizophrenia. Mutations in the MAOA gene, which is not a common one, but it causes disruption to normal synaptic function. It causes an X-linked behavioral disorder, and that disorder causes abnormal aggressive violent behavior in males. So sometimes aggressive violent behavior in males is actually genetic. However, it is triggered by environmental factors. So if the right environmental factors aren't there, this disorder won't be triggered. But between the gene and the environmental factors, if the conditions are just right, this disorder could be triggered and cause the male with this specific mutation in the gene to become abnormally aggressive or violent. It's a pedigree of a family with a mutant MAOA allele and their abnormal behavior. You can see females are the carriers. Those are the little circles with the dots in them, but males are the ones that exhibit the actual behavior. Huntington's disease is a model for neurodegenerative disorders. That means disorders of the brain when the brain just deteriorates. It's an autosomal dominant disorder, so it's a fairly simple disorder. It's one of the few rare ones. It's caused by the accumulation of a mutant protein in the brain called Ch.18, which causes brain cell death and abnormal behavior like dementia. It's adult onset. Most people do not know that they have this disease because it doesn't exhibit itself until the person is maybe 40. But once the symptoms start showing, that person generally has about 10 to 15 years to live. And you can see the MRI findings on the right in the normal brain. And you can see the brain all filled in. It's nice and fat. It has a lot of different branches. And then here you have all this space. So a person with Huntington's disease, their brain tissue just degenerates. Their brain cells just die. And it's because they have an excess of that mutant protein. The gene for HD is located on the short arm of chromosome 4, so scientists actually know a lot about Huntington's disease. Mutant alleles carry more CAG triplet repeats than normal alleles. The expansion of the number of CAG repeats causes many more copies of the amino acid glutamine to be inserted into the protein encoded by this gene. And this increase, called a polyglutamine expansion, causes the protein to misfold. It doesn't fold properly, and form aggregates or clusters of this protein, which make it toxic and kills the brain cells. Animal models. We talked a while ago about how we use mice for pretty much everything. Transgenic mice models have been used to investigate the biological and genetic mechanisms that underlie alcoholism, schizophrenia, bipolar, Alzheimer's, ALS, and Parkinson's disease. We use mice because they develop symptoms similar to the condition in humans. So we basically raise mice with these disorders so we can study them without having to, not necessarily infect, because it's not an infection, but if they wanted to study humans, they have to wait for the humans to develop symptoms of these diseases and then go to a researcher. And a lot of people with these disorders aren't going to do that. So they've genetically modified mice to purposely have these disorders to be able to study it. Transgenic mice were engineered to carry mutant human HD genes with various numbers of trinucleotide repeats. And you can see pictures on the bottom of their brain tissue. Picture A is a normal mouse brain striatum, a part of their brain showing densely packed neurons. And B is a section of the striatum from the Ch.18 mouse, so a mouse that they raised with Huntington's disease, showing extensive loss of neurons that accompany it. Potential therapies. Research has identified important proteins that play a role in the development of HD. RHES is a protein that interacts with the MHTT in brain regions where neuron death occurs. RAB11 is a protein involved in building neurotransmitter vesicles whose activity is inhibited by MHTT. Remember, too much MHTT is what causes brain cell death. In addition, other research has identified genes that lower the levels of MHTT, which is what we want. We don't want too much. One of these, NUB1, encodes a protein that significantly lowers the levels of MHTT and reduces cell death. So by identifying these genes and these chromosomes and these proteins, we've been able to develop therapies for people with Huntington's disease. Genes and environmental factors affect behavioral phenotypes. Schizophrenia and bipolar disorders are caused by many genes, each with a small effect, along with environmental triggers. Autism spectrum disorders are complex. Major depressive disorder, ADHD. These are all a combination of both genes and environment. Schizophrenia. It's called a complex multifactorial trait. Many factors contribute to schizophrenia. And it has to do with abnormal glucose metabolism. So the metabolism of glucose in the brain of a schizophrenic individual left and a normal individual right. So there's a lot of yellow, which means there's a lot of metabolism going on in a schizophrenic person. And a normal person on the right, not so much. The increased risk associated with increased genetic relatedness suggests a genetic component to schizophrenia. So the more related people are, the more likely they are to develop the disorder. Using a narrow definition of schizophrenia, the concordance value for monozygotic twins is 46%. versus dizygotic twins, it's 14%. Remember, monozygotic are identical twins. Dizygotic are fraternal twins. If they use a broader definition, concordance for identical twins approaches 100%. Both twins develop schizophrenia. And 45% risk for siblings, parents, and offspring of the schizophrenic. The higher risk with monozygotic twins indicates a genetic component, like I said. The fact that not 100% of identical twins always develops the disorder indicates there's an environmental component in addition to genetics. So the gene might be there, but something in their environment has to trigger it. Many psychiatric disorders share alleles, SNPs, and CNVs. At the bottom, I put CNV stands for copy number variant. We studied those in chapter 6. And the fact that we know this may lead to new approaches for diagnosing mental illness. It's easier to diagnose a mental illness if a person maybe gets their DNA profiled. And researchers can see exactly which allele they have and if they will, or if they have the potential to develop a disorder. Examples are schizophrenia and autism spectrum disorders. Duplications of a certain CNV is associated with schizophrenia, but deletion of that same CNV is associated with autism. So it's the same copy number variant, the same section of a specific gene or chromosome. But if it's duplicated, then the person develops schizophrenia. If it's deleted, the person might develop autism. That is figure 1811 from your book. SNP and heritability for individual behavioral disorders are the blue columns. And heritability across disorders are the green columns. The degrees of heritability are shown on the y-axis. And there are 4 pairs of disorders that show strong correlation with the same SNPs present in both disorders, suggesting that these pairs of disorders share an underlying gene. So like I said, autism and schizophrenia share an underlying gene because that same gene can either be duplicated where it causes one thing or deleted, where it causes the other. Epigenetics and mental illness. We've gone over epigenetics several times, and I put a link at the top if you need to review epigenetics. In monozygotic or identical twin studies, they've done studies with where one twin has a mental illness and the other one does not. They've done studies for schizophrenics and bipolar disorder. I actually have cousins that are fraternal twins. One of them has bipolar disorder, one of them does not. They study the differences in DNA methylation patterns and chromosomal regions associated with both the disorders. And they've discovered a gene, ZNF652, where if that gene has an increased methylation pattern, it's likely that the individual has schizophrenia. If it has a decreased methylation pattern, it's likely that the individual has bipolar. So this suggests an epigenetic component to these mental disorders. Genetics and social behavior. Social behavior is interactions between and among individuals. This is also a multifactorial inheritance where genes and environment are both contributing factors. Alzheimer's and addiction are both examples of these multifactorial disorders. You have to have the right gene, have to have the right environment to be able to develop these. Alzheimer's disease is progressive, fatal neurodegenerative disorder. Neurodegenerative means your brain degenerates. Brain lesions formed by the accumulation of an amyloid beta protein. Age is a major risk factor. This happens mostly in older people. There are two forms. A familial form, which is usually onset a little bit earlier, caused by mutations in any of the three genes associated with the amyloid beta protein AD1. But this is less than 5% of all Alzheimer's cases. So this type of Alzheimer's disease is very rare. The second form is called sporadic. This is late onset. This is 95% of the Alzheimer's cases. And it's caused by all three: genetic, environmental, and epigenetic factors. And then the table, scientists know they can list the genes that are involved in developing Alzheimer's. Now that they know the genes, now they're working on therapies. How can they fix this? Brain lesions are formed by a protein fragment that accumulates in groups known as senile plaques in the regions of the brain associated with the limbic system. The limbic system includes the hypothalamus, the thalamus, the amygdala, the hippocampus. A lot of it has to do with memory, which is why people with Alzheimer's disease forget a lot. And that's what they look like under the microscope on the left. Addiction is known as compulsive and repetitive actions known by the individual to be physically, psychologically, or socially harmful, leading to clinical impairment. Addictions develop from a stepwise series of physiological adaptations that alter brain function following repeated exposure to certain environmental stimuli. The stimuli could be a lot of things. Could be alcohol, could be nicotine, could be drugs, and it could be gambling. Compulsive gambling is an addiction. It changes the physiology of a person's brain. Family studies have revealed a genetic component to addiction. The heritability of addictive behaviors derived from twin studies. Higher heritabilities are associated with higher risks. As we said, the higher heritability, it's actually a scale from 0 to 1, actually. There's everything on that list from hallucinogens, stimulants, cannabis, sedatives, gambling, smoking, alcohol, caffeine, opiates, and cocaine is the highest. The nicotine addiction, smoking. The effects of nicotine result from its binding to the cell surface receptor. The neurotransmitter acetylcholine is important in regions associated with mood, attention, arousal, and memory. Nicotine acetylcholine receptors are a large family of proteins that mediate signal transmission at the synapses. Those are the receptors that we looked at in the GIF. The receptors are composed of 5 protein subunits. And people have different alleles that control what these subunits look like. Allelic variants of these genes and different subunit combinations create combinations of receptors with either stronger or weaker abilities to bind to nicotine. People with receptors that are strongly bind to nicotine are susceptible to nicotine addiction, whereas others with weak binding receptors are more resistant to addiction. So nicotine has a genetic component. What do your nicotine receptors look like in your cells? Those receptors are controlled by your genes and can either bind strongly, in which case people are more likely to become addicted to nicotine, or they bind weakly, in which they're not. They most likely won't get addicted to nicotine. And there's genetic risk factors involved in nicotine addiction. There's all the different genes controlling all the different receptor shapes. Now, alcoholism. As a behavioral disorder, excessive alcohol consumption has 2 important components. Drinking in excess over a long period causes damage to the brain, nervous system, and other organs. Over time, the accumulation of damage results in altered behavior, hallucinations, and loss of memory. The second component involves behaviors that lead to the excessive alcohol consumption and the loss of ability to function in social settings, the workplace, and at home. So there's a physiological component where the body and the brain changes, and a behavioral component where people can't function normally anymore. In mice, experiments show that alcohol preference can be selected for. Some strains of mice choose a solution of 75% alcohol over water, whereas some strains shun all alcohol. There's a 25 to 50% risk of alcoholism in the sons and brothers of alcoholic men. There's a 55% concordance for alcoholism in identical twins, and 28% rate in fraternal twins. And they know this because they've done adoption studies. And sons adopted by alcoholic men show a rate of alcoholism closer to that of their biological fathers than that of their adoptive fathers. Early searches for genes that influence alcohol consumption focused on genes involved with the metabolism of alcohol. The two proteins involved in the metabolism of alcohol are alcohol dehydrogenase and aldehyde dehydrogenase. Alleles of these genes cause a wide variation in the rate of alcohol metabolism, some which provide resistance to alcohol dependence. Just like there's genes that confer resistance to nicotine dependence. There's figure 18.15 at the top. That's the normal pathway of alcohol metabolism. So you drink alcohol. ADH1B is a protein that's going to convert it to acetaldehyde. And then another protein, LDH2, converts it to acetate. And the pathway moves along. So a certain allele of ADHB1 converts alcohol to acetaldehyde 100 times faster than the normal allele. And the second one, an allele of ALDH2, lowers the rate of conversion from acetaldehyde to acetate. Both of these raise the level of that middle molecule, the acetaldehyde, which causes an unpleasant reaction to alcohol consumption, headache, nausea. In populations where one or both of these alleles are present, these genotypes confer protection against alcoholism. People have bad reactions when that molecule builds up in their blood. They don't like it, so they just don't drink. Summing up the current status of human behavior genetics, searching for single gene effects were unsuccessful. Not very many disorders are controlled by just a single gene. Most behaviors are complex traits involving many genes, each with a small additive effect. Means they build up with each gene you have or with each gene that gets turned on. Genomic studies may be able to identify genes associated with mental illness. They've identified a lot of them so far. And the environment, including epigenetics, plays a significant role in behavior.